Hello and welcome to our fourth week if you're taking the Science and Scripture course, or this is the fourth science video if you're just watching at random. And this week's topic is Adam, Eve, and genetics. And this is where it gets a little harder, I think. We did in week one, we talked about 4 different views of how science and Scripture might fit together. And many times we talk about science and Scripture as being in conflict. I do think a lot of times they're independent. They're just talking about different things. I hate to leave it at conflict. I'd rather engage in the third path, which is dialogue. And my ideal, of course, is the fourth path, integration, finding a way to fit them together. We can't always work that out. And in week two, we talked about the Big Bang. I didn't personally find much difficulty there in terms of the fact that when God said, let creation happen, that it happened with a bang doesn't seem problematic to me. Last week, we talked about quantum indeterminacy, which as a Wesleyan Arminian that believes that God empowers the creation to have some freedom, that's not a big deal to me. It's really this week when we get into the question of Adam and Eve, the question of DNA and genetics and so forth, that's where I feel the real tension begin. And of course, I don't want to leave things at conflict where we have to choose the science or a particular understanding of the biblical text. My ideal is for us to dialogue because interpretation is not always solid either. And the science, of course, develops a little bit. So I think we can dialogue between the two with the goal of integration at some point. And let me reiterate. So my PhD is in New Testament. And I don't know that I was a smart aleck, but I was a kind of internally cocky. I don't think I was externally cocky, but when I went to college and when I was in seminary, I was cocky on the inside. I can figure this out. But, like when people presented, I was in a class on the Gospels once, and we were looking at the denials, the 3 denials of Peter. And you look at them, and then you look at John. John is the one that's a little difficult to fit with Matthew, Mark, and Luke if you want to reconcile them on the level of detail. And I didn't say it out loud, but inside cocky me was saying, I can figure this out. I'm a pretty smart guy. I can figure this out. And I remember I wrote a paper on it. I did an all-nighter before it was due. Again, don't do that. Learn from my mistakes. At 4 a.m., I always told myself, why did I do this to myself? Anyway, that's a different story. But at 4 a.m. writing this paper, I did Matthew, Mark, and Luke, and I thought, yeah, this is a piece of cake. Internally, man, am I smart? And then I got to John, and I came up with something. I turned in the paper. I probably got an A- on it, but I really didn't solve the problem. And of course, since then, I've come to believe that that's a wrong-headed enterprise. That the idea that we have to harmonize the gospel on a minute level, that's a modern preoccupation. That ancient history wasn't worried about that level of fine-tuning of things and fitting them together. So I've come to think that whole enterprise of trying to harmonize, was it 1 blind man? Was it 2 blind men? Was it going into Jericho? Was it coming out of Jericho? I've come to think that that's just a wrong-headed thing to worry about. That we can say the Bible's inerrant, and not try to reconcile those things because there was a flexibility to ancient history writing that we would not do it today in our way of history writing, but the Bible wasn't written today. It was written in their time. It's the standards of ancient history writing that we should be paying attention to, not our standards of history writing. Well, again, you don't have to agree with me on any of those things, but what I'm saying is that I started out my biblical studies career kind of mocking those stupid liberals, who come up with their cockamamie theories. I used to mock them, and then I began to study them, And over time, I thought, well, you know, first of all, not all these people are evil people with these long mustaches. I'm a liberal, that was probably not a very Christ-like attitude to have toward those people anyway, because a lot of them were good people. And here I was villainizing them, and I didn't even know them. And then secondly, I began to discover that maybe some of their cockamamie theories actually had some basis in the text, and that I was the one who was, we mock that which we do not understand. My point in telling you on that is to indicate that I've long since, and I went through a faith crisis because of it. My cockamamie certainty that I know I'm smarter than these people. I have it figured out. And then to actually realize, what, I don't know anything about anything, and to work through some of these things. And of course, that's when I really came to appreciate the motto, Christ has died, Christ is risen, Christ will come again. I'm good. That we can work out, don't get me wrong, we can work out a lot of details. I'm not against working out the details, but that's all icing on the cake. Christ is risen from the dead. He is risen indeed. He is Lord, he is Lord, he is risen from the dead, and he is Lord. We're good. And these other little these border skirmishes over the details are distractions, I feel, a lot of the time. Well, my point is that the things that we always tend to assume, well, the interpretation part is ironclad. Well, I'm not sure the interpretation part is as ironclad as I thought. Well, certainly, I don't think it was as ironclad as I thought it was, that I had it all figured out, when I was 18 or even 17 or even 16. I had it all figured out. Well, I didn't have it all figured out. And so, we always talk about the science being the part that needs to, well, obviously the science is wrong, but it may actually be, in many cases, our interpretation that is not very sophisticated. This is one of the I love democracy. I love America. I love the Republic. We get to vote on our leaders. I love that. We get to vote on our representatives who pass the laws. I love that. But one of the, for every strength, there's a corresponding weakness. One of the weaknesses of American democracy is we think that because we get a vote, we know as much as everybody else. There's a strong stream of anti-intellectualism in American culture. There's a strong stream in American Christianity that says, I've got a Bible and I can interpret it as well as anybody else. Well, the Holy Spirit speaks to everybody, yes. No matter whether you have a degree in Bible or not, the Holy Spirit can speak to you. And sometimes the intellectuals are flat wrong. Sometimes they can't see the forest for the trees. Absolutely true. But what I'm saying is there are actual experts who can read Greek and Hebrew and Aramaic, and who know the ancient world, and they know the history, and they know the cultural context in which the biblical books were written. And it is simply the case that they are more likely to understand the Bible for what it meant than someone who just picked up a Bible in English and did the hunt and peck method. Judas went out and hung himself. Oh, go down, do likewise. Oh, what thou doest, do it quickly. Oh, there are experts on the Bible. Okay, that's the preamble. So I want to get in. This is the science video. We'll talk about the Bible portion in our lifetime. But this is a video on Adam and Eve and genetics, and I'm going to do my best to present the science as best I understand it. Now, some of this science is fairly recent, and by recent I mean within the last 20 years. And so I don't think we should assume that the paint is dry on all of it. And so there's a certain tentativity I want to convey as I go through the science as I currently understand it. Now, in the good old days, when I started teaching back in 1997, so I started teaching at Indiana Wesleyan University I'm now at Houghton College, but I started teaching in 1997, and I had a biology professor friend. He went on to become the president of a college in Kentucky. As I recall, when we would talk about evolution, every once in a while there'd be a lunch, where we'd talk about stuff. And I seem to remember him around the year 2000 suggesting that maybe what happened is that you had evolution, and then at some point, God put a soul into Adam and Eve. That there was a male, let's call him Adam, and there was a female, and these are Homo sapiens, these are humans. They've evolved over millions of years, according to this theory. And so you have an Adam and an Eve, and God puts a soul into them. And now we have real humanity, moral humanity, eternal humanity, because now we have hominins who have a soul. Now, if you're part of this class, you know that John Walton, Mr. Walton, suggests that perhaps this is something like what actually happened, that you had maybe 10,000 or 50,000 Homo sapiens somewhere in sub-Saharan Africa, maybe 50,000 years ago, and you have this collection, and God puts a soul in Adam and Eve as representative king priests for Walton. These are two, a male and a female, who are the kind of representatives of that pool, genetic pool, as it were, that pool of hominins, and God puts a soul in them. And then you could argue, well, then for every child born to them and their offspring, God puts a soul. From then on, there's a soul that God, he puts a soul in the old divine pinball machine, and ding, puts a soul in each human being born to their descendants from then on. I mean, you could even go wild with this and say that the sons of Adam are the Homo sapiens with souls, and the daughters of men are the Homo sapiens without souls. I'm not in any way suggesting that, in the flood story in Genesis chapter 6. I'm not suggesting that. But 20 years ago, this was a fairly easy suggestion to make. As we're going to see, genetics has complicated things a little bit. Walton's theory could still work, of course. The complication is the idea that all humans today have descended from a couple that lived at the same time. That's the difficulty. Genetics has, at least at the moment, I'm not saying that it won't clear up, but at least at the moment, it seems difficult genetically to suggest that all the humans that currently exist descended from a single male-female pair that lived, say, 50,000 years ago. Okay, or 10,000 years ago. All right, so is it 10,000 years ago? Okay, let's keep going. No, it's 10,000 individuals 50,000 years ago. Okay, sorry, there you go. So you might have said 20 years ago that all the rest of us humans had descended from that original pair. Okay, now enter the Human Genome Project. Now, the Human Genome Project was ironically led by Francis Collins. We've been reading Francis Collins' classic book, The Language of God. It's one of the textbooks for this class. Francis Collins came to believe in God by way of C.S. Lewis. And he led the Human Genome Project, which was a project that tried to map the entire human genome. I've got a DNA strand behind me right now, in fact. And so Francis Collins led this, and he would suggest that the human gene pool requires a pool of at least 6 to 10,000 hominins. That is, and this is Scott McKnight, who's a New Testament scholar, in Adam and the Genome, and I'm going to let you, those of you who are especially wanting to, who are biology majors, I'm going to send you a chapter from both the book I just showed, Adam and the Genome, and also a chapter from Swamidass, who questions some of this book. Swamidass has recently written a book called The Genealogical Adam and Eve. So I'm going to send you a chapter for those of you who want to go above and beyond to read in there. But my sense is that Swamidass is the minority voice, that the vast majority would agree more with Venema. That's my sense of the lay of the land in terms of scientific experts within the Christian community, even. Here's what Scott McKnight says in that book. The DNA in current humans could not have come from a pool of fewer than approximately 10,000 hominins. That's Scott McKnight's claim, and I double-checked with him a couple weeks ago, and he would say that Venema continues to hold that position, that Venema has never varied on that particular perspective. Okay, so it has been suggested, again, from these sorts of sources, and BioLogos, by the way, you may know BioLogos is a organization that works on the integration of faith and science, and so you could also search BioLogos if you want more sources here. Swamidass, from what I understand, has had a pretty significant falling out with BioLogos. Anyway, he's not on the approved list at the moment, from what I can tell. So it has been suggested that such a funnel of these 10,000 hominins would likely have been located about 50 to 150,000 years ago in East Africa. So the current theory is that about 50,000 years ago there was this expansion of Homo sapiens out of Africa, that into Europe and so forth. Now there were other hominins around. Neander, again, you don't have to agree with any of this. I'm just trying to present what I understand to be the position that the majority of experts actually think at the moment on these sorts of things. Again, maybe they'll change their mind, but at the moment, this seems to be a fairly strong consensus among experts. So in Europe, we have Neanderthal. You've heard of Neanderthal. There were other humans that were more prevalent. So for example, there was a different group of closely related hominins in Asia. So Neanderthal tended to be more in Europe, but over in Asia, we had a different hominin that was already out of Africa, and their name were Denisovan, if I remember correctly. So the idea was the Homo sapiens came out of Africa about 50,000 years ago, that there was some interbreeding, I'll say breeding, so there is a certain amount of allegedly Neanderthal DNA in people like me who came out of Europe, and there would be a certain amount of Denisovan DNA in Asian humans and in North American indigenous peoples and so forth. Anyway, it's all very interesting. Again, you don't have to accept any of this, but this is what the majority of experts currently think about these things. So the Human Genome Project has basically suggested that there is, and I don't know the details, some of you may actually know this, some of you I think are taking genetics right now, that the argument is, and again, I'm going to send you a chapter from Swamidass because I think he pushes back a little bit on this, is that we need a fairly large pool of human beings in order to have the amount of diversity of genetic material that we have. Now, you could argue that God created us to look like we had that much diversity, or you could argue, because it's generally considered that the greater diversity, this is the piece I'm going to send you from Venema, talks about how the Tasmanian devil is in trouble because it's lost a lot of its genetic diversity. And so a species that doesn't have that kind of genetic richness is more susceptible to elimination by disease, because when you have a diverse genetic pool, if there's a pandemic or whatever, it may be likely that part of the species is wiped out, but the part that had that richness of genetics that was immune or resistant would go on. But if you have a genetic pool that isn't very diverse, then all it takes is the silver bullet of a disease and it's all gone. And so the genetic richness of the human genome, according to the current hypothesis, is that it must come from at least 10,000 hominins. But you could argue that God created it rich. I'm not arguing for this, but you could argue, well, God made us, God miraculously made human DNA genetically rich, so that we would be a healthy human being population. You could argue for things like that. Okay, that's invoking the miraculous. You know, to use the model I've presented, this is God intervening in the normal cause-effect processes of the world in order to do something, and now for something different from God. Okay, well, let me back up a little bit and talk about the science of genetics. So it was only in 1944. I mean, really, this understanding is a fairly recent kind of thing. It was only in 1944 that it was determined that this DNA, this double helix, you know, stair ladder, was the keeper of our genetic material. That this is the map to my blue eyes and brown hair, and my fact that I have earlobes and jowls. Anyway, in 1953, James Watson and Francis Crick identified this basic structure of DNA, that it's a double helix, it's a twisted ladder. This is, by the way, kept inside, not only in our cells, every cell has this complete map, but in our, not only in the nucleus of our cells, but in the nucleolus of our cells when it's not dividing. Each rung in the ladder is a base pair. So it's a mixture of four different amino acids. So you have adenine, thymidine, guanine, and cytosine. And these tend to pair up in certain predictable ways. So only certain ones, you know, connect with other ones. But there are like 3 billion pairs of this thing. There was a race, as you can read about it in the language of God. There was a race to map this thing. There was a tension between the universities and the government that wanted it to be public. And then you had some enterprising capitalists who wanted to basically own it, to where, you know, if you want to know about the gene pool, you got to pay us money. And of course, I think it was much more of a virtuous possibility for it to be public, and so it is. They won out, and they thought it would take a long time because we're talking 3 billion. But what we found is, what they found, I haven't, is that there's a lot of emptiness in there. There's a lot of kind of junk, what we call junk DNA. More on that in a second. I'll talk about that in a second. But this is taken from the U.S. National Library of Medicine. That's the picture I have behind me, and you can see that guanine and cytosine pair up, and then adenine and thymine pair up. So this is the basic structure of deoxyribonucleic acid. So let's get to the heart of the matter here. So one of the reasons why it was possible to map the DNA so much more quickly than they thought is because, as it turns out, and again, this is where the science is currently. It may turn out that some of this is not junk. There's some people who really don't like talking about junk DNA, but it appears that a lot of DNA is not functional. A lot of human DNA doesn't do anything. And so our first impression is, again, some of you are studying this right now, so I'm going on this material. My impression is that if you were to design DNA from scratch, you simply wouldn't design it the way it is designed, because there's an awful lot of DNA that doesn't actually do anything. It's not streamlined. There are apparently stretches of what we call junk DNA that doesn't do anything. In fact, only about 1.5% of that billions of pairs is actually useful. Only maybe 1.5% of DNA actually tells us anything. Now, what's interesting is these long stretches correspond to stretches in other organisms that actually do function. I don't know if you can see the argument going here. The idea is that if all human life has evolved in a kind of tree to where you had, to where I have just a much more developed form of DNA than a bacterium. And therefore, you would expect there to be a broad set of correlations of human DNA with a bacterium. And you would expect there to be a broad set of correlations of human DNA with a mouse, or a broad sense of correlation between my DNA and a bird. But then there would be different parts. I would have more parts because I'm more complex, but also you would expect there to be, or at least we wouldn't be surprised if we found long stretches of my DNA that correlates to some of these bird stretches or mouse stretches, but those stretches don't do anything for me, but like wings, I don't have wings. I don't know, maybe I don't know exactly the particulars of where they are on the DNA, but we have long stretches of wasteful DNA in the human DNA that correlates to useful DNA in simpler organisms. You see the argument here? The argument is that if God were to create human DNA de novo, out of the blue, just completely just for humans, it wouldn't need to be nearly as long. God could just focus on that 1.5%. On the other hand, if God, and again, I'm assuming if this happened, it's theistic evolution, or maybe we could call it evolutionary creation. I'm assuming that if God God is the one who used evolution to create humanity. So I want to throw away right now the dualism of either it's atheistic evolution or it's 6-day, 24-hour day divine creation. That's a false alternative. There is this other alternative that God was deeply involved in the evolution of life. In fact, when I was a teen, we said, well, where are the transitional forms? That's what we used to say. And so theistic evolution, or I would prefer to call it evolutionary creation. You could say that when God got to the gap between amphibians and reptiles, God says, zap, the idea of God intervening in the evolutionary process where I'm going to nudge it this direction. Again, I'm not saying that's the way it happened. What I am suggesting is there are probably more possibilities here than your typical arguments that you hear. That there are all sorts of possibility for nuances. So, for example, perhaps God said, again, I'm just doing thought experiments here. I'm not saying how God did it, but let's say that God said, I'm wanting to have an atom here. And these dinosaurs, it's not going to work. So here's what I'm going to do. I got this asteroid. I'm going to let it hit the Earth. And pretty much all of the surface living creatures of the Earth are going to die, including the dinosaurs and everything. But there's this little mole-like creature that lives under the surface, kind of a mammal. He's a nothing now. He's an insignificant nada. But after things cool down, after this asteroid, after the dinosaurs are gone, everything, I'm going to use this mole to create the line from which I will eventually get an atom. And I'm going to zap it at just the right time. I'm going to zap the mutations. God is mutation maker. Got to make a song, mutation maker. No, I'm joking. Anyway, again, I'm not saying God did it that way. I'm just saying that there are more things, in the words of Shakespeare, there are more things in heaven and earth than are dreamt of in your philosophy, Horatio. Again, I'm not saying how God did it. Maybe God gave us a parent age. We talked about that theory, that God could have created Adam and Eve with a parent age, to where their DNA had a parent diversification and so forth. But it is curious that human DNA has so much, 98.5% of our DNA is pointless, apparently, but it corresponds to material in the DNA of earlier DNA that actually did things. So here's another one. There's no efficiency in the base pairs. The base pairs do not efficiently code for certain amino acids. There's redundancy. So, for example, both the code GAA and GAG both code for glutamic acid. And so why do that? I mean, if you were going to design this thing efficiently, you know, in the good American way, you'd just do one pair for one acid and so forth. So there is this kind of inefficiency to the DNA molecule and the way it works and the way it performs, which again would completely fit this kind of a process that God, as the great orchestrator, you know, zap, da 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 da, zap, da 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 da, zap, you know, where God is letting the creativity of the creation move along, and God enjoying the beauty of this world that in a sense he's created to create itself to some extent, but he has a purpose because he wants to get to Jesus. And so, you know, he and he's very patient, right? A day is as 1,000 years. And again, I'm not saying that's the way it happened. I'm saying that I can imagine a world in which God did that. Now, next week, we're going to talk about evolution in the fall. And I've said several times in the course that where it really gets difficult for me is with this question of how sin entered into the world. So I'm not suggesting that this is a theological cakewalk. I'm not. But I am doing the thought experiment. I'm doing the dialogue. And this is the science part of the dialogue for this week. So now we get to mutations. You know, it's interesting that Darwin didn't know about mutations. He had no mechanism. C.S. Lewis talks about this in a sermon he did, the World's End. I forget the name of it, but basically he talks about how the world in the 1800s was looking for a Darwin to tell them that progress and evolution was the way it happened scientifically. It's not that Darwin came along and then everybody came to believe in evolution as a human cultural thing. Progress was the name of the game, and Darwin fit right into that. Darwin had no mechanism to explain how his theory of evolution took place. It was later that we discovered the idea of mutation. Of course, most mutations are harmful. And a lot of times when I've listened to debates against evolution, this is one of the things that is often said, that mutation usually harms something. Mutation doesn't usually make things better. Well, we have 23 pairs of chromosomes in which the DNA is found. Francis Collins, early on in his career, discovered that the deletion of just 3 letters of the 6 billion letters is responsible for cystic fibrosis. And so, of course, one of the fears of genetic testing is that we will abort babies that are going to be born with various maladies and things like that. But definitely it is true that, well, we've found that certain parts of this DNA correlate to certain maladies and so forth. At the DNA level, all humans are 99.9% identical. So we can't pull up, again, no one on this, watching this video would believe this, but there were, of course, those who used to argue that perhaps somebody of a different race came from a inferior DNA strand, you know, or something like that. But we are all, as humans, very, very, very identical. Now, what happens with mutation is that there's about 1 error every 100 million base pairs per generation. So that means that each person has about 60 mutations. Well, you think if I have 6 billion letters in my DNA, 60 mutations is not very much at all. And probably those mutations are going to take place in the junk part, right? For them to do something significant, they would have to take place in the 1.5% of my DNA that actually does something. And so but mutation is a constant. And of course, it has a much bigger effect, I would say, on the virus level. This is one of the things that I've heard people say about the coronavirus. I'm doing this video in 2020. Well, some have suggested that the strand of coronavirus, COVID-19, that came from Italy to New York was a slightly mutated and more virulent strand than what had originally come out of China, Wuhan province. So, and of course, some people were concerned about a vaccine. Would a vaccine be able to stop mutated versions of the virus? But from what I understand, most of the vaccines don't target the virus per se, but the attachment mechanism of the virus. So all of these coronaviruses attach, or this particular strand of COVID viruses attach in a similar way. And so the vaccines are not targeting the genetic material that's the virus per se, but the attachment mechanism, so that it will work even if there are variations. But on the virus level, mutation can change things very quickly, I suspect. Somebody my size, it's going to take a little bit more work to change it. Well, there's a similarity of sequence, not just in the meaningful DNA, but in the junk DNA. So the chimpanzee, which allegedly is our closest genetic relative in the animal kingdom, there is 100% similarity between the sequence of meaningful DNA for me and the sequence of meaningful DNA for a chimpanzee. Now, even in the junk DNA, there's a 98% correlation between the junk DNA of a chimpanzee and the junk DNA of a human being. Now, the further down we get in this list, the less correlation. But I mean, a dog, I like to think that I'm far superior to the dogs I have. I have some dogs, and especially the beagle. I am far superior to this beagle. Don't get me going on beagles. But 99% of my DNA is in the same order as the DNA in a dog. 99% of my DNA is in the same order as a mouse. 75%, I like to think of myself as being quite different from a chicken. 75% of my DNA is in the same order as a chicken DNA. 60%, man, a fruit fly. Are you comparing me to a fruit fly? 60% of my DNA apparently is in the same order as a fruit fly. A roundworm, okay, thankfully I'm not like a roundworm. 35% of my DNA is in the same order as a roundworm. Now, the junk DNA, 52% of my junk DNA is in the same order as a dog. 40% of my junk DNA is in the same order. So this is the stuff that doesn't do anything. And of course, Francis Collins singles out this mouse similarity in his book. So I hope you'll read that. So again, you could say, well, why reinvent the wheel? God decided to kind of follow the same concept. Maybe. This reminds me a little of a question in biblical studies called the synoptic question. Why are Matthew, Mark, and Luke so similar? What we find is that Matthew, Mark, and Luke have very similar wording to each other in some places, and not just the words of Jesus, the storytelling. They have very similar order to each other. And so the overwhelming majority of biblical scholars believe that Mark was a common source behind Matthew and Luke. It's the same kind of reasoning. And with the synoptic problem, or the synoptic question, you can say, well, maybe God just did that. Maybe God dictated it to where same word, different word for and. Same word, this is present tense, this is past tense. Same word, oh, these two words are flipped around in their order. Maybe God just was having fun. Maybe he nudged Gabriel and he says, in the 21st century, this is gonna drive him crazy. Maybe God did that. Maybe he said, I'm going to design the DNA so it looks like humans have evolved from simpler animals. But it's just the way I'm doing. I'm having fun with it. I'm testing them to see if they have faith or not. And those who have faith will say, even though it looks like evolution happened, anyway, I just wonder why God would do that. Why would God do that? And of course, if it's necessary for us to believe that, of course, God can do anything. God is a God of miracles. So the question is it necessary? Is there anything that compels us to not go with the way things seem to be? Is there anything that would require us as Christians to say, even though it looks like there's a kind of relationality here, by faith, I'm going to go with the blind faith option. That faith is irrational. Faith is blind. It doesn't make any sense, and therefore it's true. I've never really liked that version of faith. I like the version that faith is reasonable. It's not that the evidence necessarily proves faith, but the faith is rational. It's reasonable to believe in faith. So, okay, we'll get to the theology questions eventually, not in this video, but we will get to it. Now, here's the content similarity. 96% of human DNA is similar to that of a chimpanzee. And then again, the lower we get on the complexity scale, the less the similarity of the DNA. So I have 60% similarity to banana, believe it or not. You heard it here. Shank is a banana. So again, these are the reasons why many have concluded that evolution is the most likely way to explain how human beings have and these other animals and plants and so forth have come about. Okay, so here's some more arguments, and you'll find this in Collins. And if you want more materials, I've bought lots of books. It's my greatest weakness. There are AREs, not ROUSs, rodents of unusual size, for those of you who like The Princess Bride, but AREs, ancient repetitive elements. These are segments of the DNA that in a human DNA don't do anything. They do something in other animals, but and some of them are decapitated. By that I mean the full DNA element is not in humans. So let's say that, I'm just going to make some stuff up. Let's say that we had four components in a pig that in a pig did something, but and three of those four components are still in human DNA in roughly the same spot in the DNA. But you need all four. Let's say you need all four for it to work, whatever it was. But we only have three of them in humans. So they serve no function whatsoever in human DNA. But they're in the same spot as they were in the pig in a slightly decapitated version. So why would God do this? Now, again, far be it for me to tell God what he can do. If God wants to have fun, man, I'm going to drive him crazy in the 21st century when genetics pops. Hey, Gabriel, it happened. Come over here. Collins is going crazy. Anyway, I just don't know why God would do this. And of course, the simplest explanation, without being too simple, is usually the right explanation. And the simplest explanation is that this is a remnant of evolution. These are in the same positions, but they're non-functional. So here's another example. The gorilla and the orangutan have 24 chromosomes. We, as we just said, have 23. Now here's the thing. Our second chromosome looks like the chromosomes next to each other in the gorilla have been fused together. So again, this is really, what a strange coincidence that we happen to have in one of our DNA what is two strands in a gorilla, but it's all put together. Why would God do this? Again, it's God's business. God can do whatever God wants. But it's the simplest explanation is, of course, the evolutionary one. Here's another one. There are certain pseudogenes that we have. They look like genes. They don't function in us, but they do function in a chimpanzee. And here's the thing, one of them, because ours doesn't function, we have a lighter jaw that may allow us to have a larger brain. So again, I'm not saying this is how it happened, but you can create a novel where God says, and now I'm getting ready for Adam. And so I'm going to take this gene in the chimp and I'm going to make it not work so that I can make way for a bigger brain in Adam. Again, I'm not saying that's how it happened, but I'm just saying that you can see that, well, they're not crazy. Evolutionists aren't crazy, is what I'm getting at. It's not, I remember when I was in I think it was in the 8th grade. I thought I was very clever. I came up with this argument by myself, as far as I can tell. I said, if evolution is true, I was vanquishing my 8th grade science teacher. If evolution is true, then and we evolved from an ape, why do we still have apes? Man, I'm clever. I thought I was so smart. Actually, I was talking to someone else, and he was a very, very nice man. He said, well, the thing is, evolutionists don't technically believe that humans evolved from apes. Evolution technically believes that both apes and humans developed from a common ape-like ancestor. So as it turns out, I didn't even know my evolution. But again, and I said this the first day of class. I would hate for a Christian young person to lose their faith over this. I really would. There are other alternatives out there. Francis Collins is a believer. Dennis Venema is a believer. Scott McKnight is a believer. There are believers out there who have found a way to integrate this with their faith. Now, they may be right, they may be wrong. I don't agree with Collins on everything, and so forth. But what I'm saying is there's no reason, in my opinion, for a young Christian, especially somebody who goes and does graduate work in biology or whatever, there's no reason for a Christian to lose their faith over this issue. Well, my last slide for this week's lecture. Genetic variability. I started off this video talking about the idea that perhaps God, when Adam and Eve got to that stage, he put a soul into the divine pinball machine and put a soul into Homo sapiens, and that all humans have evolved, all humans have descended from Adam and Eve with a soul. Here is the challenge to a single human pair being the parents of all living beings today. And this has to do with the connection between the number of variants in a population and the size of a population. Like I said, I'm going to send you this chapter from Swamidass where he questions this logic. And so I mentioned the Tasmanian devil earlier. It has little genetic variability. And basically, scientists believe that there was a bottleneck a few hundred years ago. And because of this genetic bottleneck, there is a cancer running through the population of the Tasmanian devil. And this cancer, again, there's not enough genetic variability for the Tasmanian devil to survive this. And so what they've done is they've, as I understand it, they've isolated Tasmanian devils without the cancer, so that they can hopefully recreate a gene pool after this cancer makes its way through the population. But apparently, the human genetic variation is big enough for again to predict that there had to have been at least 10,000 hominins from which this variety came. And there are two different methods that are used to talk about this, to arrive at this. One is, how diverse are human alleles? Again, the thought is that this diversity of human alleles suggests an ancestral population of about 10,000. And then another method that comes up at the same result is to look for crossover chromosome events, which again suggests that how many crossovers there are within the chromosomes, which again suggests an ancestral population of about 10,000. Well, I'll give you some reading that will stretch, but the long and short of this is the current thinking, and again, maybe this will change. The current thinking is that we need a larger population of Homo sapiens in order to arrive at the diversity that we have now by natural means. Again, like I said, you could say that Adam and Eve were created with apparent age, which could include apparent diversity of their DNA. But from a natural standpoint, if God doesn't, we don't, here I am, I'm intervening in history. If there's no intervention, miraculous intervention of God in history on this particular score, then the current thinking is it would take about 10,000 hominins to arrive at the current diversity within our genetic pool. Well, to be continued. We're going to continue talking about this. I've tried my best to present to you the science as best I understand it. We will move forward with theology both this week and next week, talking about how dialoguing between this and faith, and seeing if we can reach seeing if you can reach as an individual some degree of integration between the two.